Spotlight, lectures and performances on and around Albany State University.
Allowing me today to come and talk with you about my research. All of you, let me just summarize for you what actually I have been doing for the last few years and what I'm interested on this topic. All of us here, we know what's chlamydia. We know that chlamydia is one of the most worldwide, US, China, Asia, Africa, the most common sexually transmitted type of, bacteria. In US, between 4 to 5 million every year, especially on this age. You are sitting here, you get to be very careful. This is the age where, chlamydia mostly infected people. What we try to do, we try to do something that a little bit different because we are the first people to do it. To use nanoparticles to try to work on vaccine for chlamydia. But the new thing here, we are trying to introduce the nanoparticle as a vaccine delivery. This simply what I have been doing through all this year. I'm going to tell you about this in a little bit of detail. This is just background on chlamydia. We know chlamydia is bacteria, and bacteria, all of you know here, is divided either into gram-negative or gram-positive. Gram-negative is most resistant to any antibiotics due to the extra, we know that, cell wall found on the outer shell. They are intracellular. In addition to gram-negative type of bacteria, they only survive inside the host, just like a virus. For a long time, scientists, they were thinking that chlamydia was a virus because of, there is a lot of similarities between virus and chlamydia. But later on, they able to, to identify it as intracellular type of bacteria. Have a very complex life cycle. It's completely different from other type, of bacteria, whether gram-negative or gram-positive. It has two type of, developmental cycle before it can, grow and develop. Chlamydia, we divided according, to very recent type of classification into 4 type of species. C-track, this the one that I working on vaccine for it, but we have another 3 different type of species: chlamydia, pistacea, and pneumonia, or pneumonia. This is the one that causing pneumonia, especially in young children, and C. pecorum. As we said, the most common sexually transmitted type of agent all over the world, here everywhere in the world. This is the life cycle of chlamydia. We talk about it has very complicated type of life cycle that completely differ from other type of bacteria. This is the life cycle. It goes through this 2 type of bodies. We call them elementary or EB, elementary body and reticulate body. The infectious body or agent called elementary body. Elementary body is smaller in size. An infectious agent, that's the one that's going to start the infection of the host cell. But immediately after enters the host cell, is going to transform itself into another type of body called reticulate body. Elementary body is smaller in size, infectious agent, but on the same time metabolically inactive. As soon as enter inside the host, it cannot do anything unless if it transforms into reticulate body. Reticulate body is very metabolically active, can divide, can grow. Within this 12 hours, it will go and, reticulate body will be divided into 2. Here, this is the persistent form. In case, if the immune system is good and strong, you see chlamydia here is acting differently from other bacteria. It will go and persist. It will stay like a dormant type of bacteria without any type, of activity till that, risk of our immune system, cleared. Then it will start initiating and activate itself into a different type of form called that reticulate body. Then it will go back, transform to elementary body. Elementary body now, it will disrupt our host cells and get out and infect a new type of cells and initiate this cycle again by reinfecting another cell. This is the type of life cycle of chlamydia. It's very complicated. It's not compared to other. Transmission mostly through type of unprotected type, of sex, and infected from infected mother to her infant. These are the major type of transmission. Epidemiology of C-track. We have to, to recognize the importance and, risk of this bacteria. Let's just have a look at this epidemiology. This in US. Just here in United States, according mostly, I got it from CDC site. Okay, the burden of C-track. Okay, we have seen the epidemiology of C-track. C-track is very, although people, a lot of even maybe in terms of even country wise, they don't take it very serious because it's not a life-threatening type like AIDS, or malaria, or it's something that do with your fertility. They think that is not something, even if you get it, you are not going to die of it. Why should we spend a lot of billions of dollars on, developing vaccine or? But let's see the burden of C-track. Between 4 to 5 million in US each year, they get the infection. 1 million of these, especially females, as we just said, 200 of these will, PID, they are getting PID yearly, and this PID or pelvic inflammatory disease, mostly due to C-track and other, STD type of, bacteria. But 2 out of these 200,000 women, about 50,000 out of 1 million get PID, 200,000 normally hospitalized every year. And out of this, 50,000 become infertile for their life. They cannot get kids. It's very high rate of, and the most important thing of here, this disease is asymptomatic, doesn't have any type of symptoms. That, the importance of this. Studies again here in US, because we are here, I don't want to show China. China is very high, 2.5% in age between 20 to 64. Very high, because China population is very high, about 1 billion. That is 2.5, it's very high. But this here we are in US, we are focusing on United States. 4.2%, very high rate for, if you are your age between 18 to 26, you get to be very careful. Okay. In China and Africa, about 15 to 45 million people are infected with, with very high rate. More than $10 billion is spent every year on just, on chlamydia and some of the diseases associated to chlamydia, like PID or pelvic inflammatory disease. A lot of money being wasted there in chlamydia. Okay. Asymptomatic disease. This one of the, important thing about chlamydia. It doesn't show any symptoms. You carry it, move it, you live very, you don't think that you are carry it. But due to this asymptomatic type of disease. Female, 70 to 90% of female don't show any type of symptoms. They don't know whether they have it or not. No symptoms. 30 to 50% of males, they don't even know that they have. Still you can have it, I can have it, and I don't know whether, until if I go and test. Okay, that means this is the, the burden of chlamydia. 4 to 5 million in US alone, 90 million every year worldwide, Africa, China, America, and Latin America, and all these continents. 90 million a year, $10 billion spent a year, asymptomatic type of disease. What do we need to do? Asymptomatic type of disease. The only solution here, vaccine. We can treat it with antibiotics. There is 2 antibiotics. It will clear it from your, within few days. But the problem, again, is asymptomatic. You don't know when you are going to catch it again. And if you want to go and screen yourself every day, there is no country in the world they have the ability, even US with this very strong type of economy, they said they cannot do a very efficient type of control, control, what do you mean? Control to screen everybody. If you don't want to develop vaccine, you get to screen everybody. And to screen everybody, you don't know what's going to happen next week. If you screen it today, it doesn't have it. Are you going to watch them throughout their life? That means there is no solution for chlamydia. We know is not a very risky type of disease, but the only solution for treatment of chlamydia is vaccine, to develop vaccine. And this is the area where I work on. we work on vaccine, and why we need vaccine due to all this. Antibiotic cannot affect already established type of infections. If the infection get into you, already this hiding, the bacteria can hide itself in inclusion body and it stays there dormant. Antibiotic cannot affect them. Secondly, current control programs are not affordable. No country in this world can afford do a control and a type of testing people, all the people. Asymptomatic nature of the disease. Doesn't have any symptoms. You don't feel nothing. You are just normal like any other. Enhanced HIV and even a cervical cancer. It has something to do with HIV and cervical cancer. Economical burden, a lot of billion of dollars spent there for chlamydia and associated type of disease. Okay, vaccine. People between year 1960 and 1970, people from United States, especially University of Washington in Seattle, they really work a lot on developing vaccine. And they tried this in some Asian countries and Africa. They took this and they went and vaccinate, immunize people there. But after the vaccine work, but of short term, it will protect you for 6 months. After 6 months, you will enhance your disease. The 4 group working together, they think that because during that period of time, everybody know that it's very easy to develop vaccine out of the lives, a type of bacteria or attenuate it, kill it, or But all these groups, they fail to develop vaccine because their vaccine just protect this patient. Mostly they use it for blind, people with, because chlamydia also, in addition to STD, they cause also, eye problems like trachoma and all this and other type, of disease. Then what they did, they tried in people that mostly they have problem of, blindness. But after a few time, these people, the disease enhanced on them. And they realized that this vaccine cannot work. After that time, people, they start looking for other type of antigens, not the cell. Now they forgot about using the whole bacteria, whether it's live or dead or attenuated. Now they start looking for a vaccine. We know that vaccine mostly, you get it mostly from the organism itself. The organism could have a protein or antigen that as soon as get into our system, our body can produce a lot of, of antibodies and type of cells that can recognize and destroy or clear the infection. Now people, they start looking for antigen, that very strong antigen that can, be able to clear chlamydia. They found this protein. We are working on it. Overall, the results show that the vaccine that produced in human and non-human primate are of short duration and exacerbate the disease. Then they found this. Any option, they found this MOMP. MOMP was discovered about 20 years ago. This is a major outer membrane protein, MOMP, MOMP, major outer membrane protein, found in the outer membrane of the chlamydia and representing 60% of the protein found on that outer membrane protein is MOMP. A major about 50 to 60%, and it has about 1182 base pair and about 390 amino acids. It has 9 types of domains. We know we did this in cell and molecular biology. We talk about when we talk about proteins. Protein, some big proteins, they have divided them into sections. We call it domain. And this protein has 9 domains. 4 of the domains are variable. What do we mean by variable? C-Track has about 18 different strains. All these 18 different strains could have their variability in this protein on this variable 4 variable domain. They just change some sequences of amino acids there, then it will give a different type of protein. And they have 5 domains that constant. All this reservoir or strains of Chlamydia trachomatis, they have 5 similar sections or domains of that MOMP. The gene for all these strains, 18 strains of reservoir, is similar 84 to 97%. What that means, this has very significant implication. We have 18 reservoir. if we're able to develop vaccine for one cell or a strain, that vaccine, is it going also to be efficient on the other strains? Since this similarity in gene between 84 to 97 is likely, if you develop one vaccine for one strain, that vaccine could also work for all the 18 strains because of these similarities. Highly immunogenic protein, MOMP, the protein, major outer membrane protein, have been discovered that, as soon as you give it to, any type of animals, the animals start producing antibodies against it. That's why people, every chlamydia vaccine people or a group of people, now they are targeting MOMP for last 20 years. But so far, there is no any, even promise or, hope to develop vaccine out of MOMP, because they have been people trying it and it's failing. But this is the amino acid sequence of MOMP of C-track, and we said it has about 1182 type of base pair and 390 approximately amino acids. And it has about 9 domains. 4 domains are variable. Mostly the variable domain are here, the domain that facing the host. These are the, and this is the immunogenic type of area, the area that, exposed to the host, the one that is going to interact with our, cells, the variable domains. That means if we need a vaccine, like what we did, we select an area from here. This area, we think that, this could be the immunogenic area. And because I use truncated type of, I truncated this peptide or gene, and I selected a small area, then we develop a peptide out of that. Okay, my work. My general objective just to try to work on designing vaccine for Chlamydia trachomatis. That's the general, goal for this work. What our rationale for that? We are thinking that recombinant MOMP, either alone or if we combine it with other type of delivery system like nanoparticle. My second part will talk about delivery system that the nanoparticle, they are going to elicit very strong humoral or antibodies type of, response, and also cell mediated. Here we are talking about T type of cell or adaptive type of immunity, because chlamydia is intracellular type of bacteria, and we know that intracellular type of bacteria cannot be cleared with our innate system. We know that our system is divided into 2 immune system, innate and adaptive. Innate system cannot clear an intracellular pathogen, such as viruses, some bacteria that live inside our host cells. It's very hard that our, mucosal secretion, our skin, our, some chemicals that produce in our stomach, these are innate type of system, cannot really clear. That means we need any type of vaccine has to activate this antibody production and activate cellular immunity. Here we are talking about T helper cells and cytotoxic T cells. Okay, our objective, we divide it again into 2 parts. The general objective. The first objective, we have to clone that truncated, as I told you. I selected a region within that big MOMP, because this is what we call subunit vaccine. Because the lab I did my work on it, they tried the whole protein, and they could not get a very good result. Then when I came doing this work, they asked me to go and select an area of this gene, and then to clone that gene and get a peptide out of that. Then I just went to the literature reading. Some people, they did work, we call it just serological type of work. They can identify what we call epitopes. Epitopes is a small region of the peptide that very immunogenic, can easily, antibody can bind to it. I went to the literature and I got an area that the literature saying this area is, very good area for, but nobody did that region as a separate region, just the, Okay, the second objective is expressing and purifying the protein. After you clone, you get to, to make sure if your clone cannot produce protein, express to produce protein, that means you didn't get anything, because, the protein is the one that's going to work as vaccine and not the clone. Okay, the second objective here, to express and purify that gene. Okay, the whole gene of MOMP, from here, about 1182, I think, base pair. I selected this area from here to here from the literature, and this is the amino acids between amino acid 187 to 344. Then the first objective is cloning. How we did the cloning? Simply, we have this whole MOMP gene in the lab that I work on. They already, extract that gene from the chlamydia. We have it there in stock. When I came, I just need to design primers for the region I selected, to clone that region. When you design primers, you run PCR. PCR is going to cut for you that region, you have the primer for it. Then, after get the region, you have to insert that to clone that region in something called vector. Vector like a carrier or vehicle that can carry this gene of interest into a bacteria, then bacteria can clone it and multiply it for it and express it and produce protein. This is what we did. We cloned that gene here in a vector called PET32. Then we express the protein in a bacteria. Then we get, most cloning now, especially if you are developing vaccine, mostly now done in bacteria, because bacteria, they replicate and grow very fast and produce for you a lot of protein within short type of time. We did this, express this and purified, then we get the purified protein. This is our result for this part. This is showing that we are successfully, we get that PCR, that gene. This is the gene fraction that I'm interested on. This is the gene fraction that I'm thinking has a lot of B and T, a type of epitopes, and that can enhance and produce a lot of antibodies as soon as interact with the host cell. After you do cloning, you get to prove that that gene, the bacteria can take it and can clone that, be transformed into bacteria, and future can give protein. This here again is a proof for us that our clone bacteria able to to take the clone, and this the gene here very clearly in all the clones. We use from 2 to 9, about 9 clones. All of them, they show our genes, on them. This is another important, result. Then the second result, now you have the gene. That gene, is it going to express itself to produce the protein? Here, did they prove that that gene also can express itself and produce the protein? Because that small protein is about 37 kilo Dalton, if we calculate that, about 166 amino acid and 571 base pair is, it will come to this 37 kD. This proof that we have the protein. Okay, the proof of the protein doesn't mean that you get the chlamydia protein. You have to prove it with Western blot, because Western blot, you use antibody. Antibody, very specific to that chlamydia peptide, has to bind. And did they prove that? We get exactly the chlamydia peptide or protein, because antibody able to to bind to it, and we show it here in Western blot. Okay, the second objective. Now we have the peptide. The ideal way, as soon as we reach this stage, we don't have to waste time here. We're supposed to take this peptide immediately and try it on mice. This is the ideal way. But a lot of time, sometimes a lot of researchers or lab, they don't allow to go directly to mice, because mice is a long and very tedious type of work. You can spend a whole year or 6 months trying a peptide, and at the end of 1 year or 6 months, if the peptide is not working, what are you going to do? That resources and time has been just, wasted. That's why our, strategy is here, why we didn't go to mice directly. We went to in vitro type of studies. And a lot of labs do the same thing. They test this, type of vaccine in cell lines. And then cell lines, they can give you idea whether your vaccine has the potentiality in future to go into mice or not. And this is what we did. We tried this peptide into mouse macrophages. Macrophage, are cell of our immune cells. They can give us idea whether they can respond if we put this in future in mice or human. Okay, we test them on macrophage to see their ability to induce this macrophage to produce what we call cytokines. Cytokines are glycoproteins produced by our immune cells, normally to regulate very important type of activity related to our immune system. They help our immune system to just get rid of the infection. And they can give us an indication whether this peptide in future can work or not. And as I told you, the very important thing here for chlamydia vaccine, you need to induce this. If the vaccine cannot induce this cellular immunity or adaptive immunity, especially T helper 1 type of immunity for chlamydia, is not going to work. Because chlamydia is an intracellular pathogen and need adaptive immunity. Again, innate immunity cannot work. That means any vaccine has to induce some of cytokines. Some cytokines, they are indicator for us that they can regulate or activate the adaptive immunity. The second part, after this peptide prove for us that it can activate these macrophage cells to produce cytokines, we said, okay, this is unusual. That means this peptide is good. And we look at the literature. There is not a lot of work even in whole MOMP have been done on mechanism. There is some part of, they didn't try to tell us the whole story of MOMP, because MOMP is 20 years or more discovered, but what the mechanism of MOMP, inducing this type of cytokines and production, nobody done a whole story. I try to tell the story of MOMP, and this paper is already, we sent it to Blue Swan like a few months ago, and they told us to do some work. But here's the whole story of MOMP. Okay, the rationale here, recombinant MOMP will stimulate J77 macrophages to produce for us very significant type of cytokines. These cytokines are very important for adaptive immunity. And also, we think that this peptide we generated in the lab is going to go through toll-like receptor 2. I'm going to talk about them. And also another pathway called MAP kinase pathway and NF-kappa B. All these are, important mediator in the whole process of cytokine production and our immune response. Okay, immunology of C-track and role of cytokines, because I'm going to measure cytokines. What cytokines going to help us in understanding the whole process, or the immunogenicity of the peptide. Infection with chlamydia normally produce a lot of and various pro-inflammatory type of mediator and cytokines, and that promote the production of interferon gamma. Interferon gamma is the cytokines. Interleukin 6 cytokines, interleukin 12 is cytokine. These are very important type of cytokine that mediate and activate T helper cells, and some of them activate inflammation, some of them, but they are good indicator that whether the peptide is going to work or not. Most of these are T helper cells type of cytokines. These cytokines trigger inflammation. We know that inflammation is a process normally our tissue and cells normally do it as trying to to clear the infection when we get injured or infected. Inflammation. We did this in immunology. Redness, heat, swelling. We call this inflammation. This inflammation is just a response of our immune system to that any type of infection. Okay. These cytokines trigger inflammation and promote the recruitment of immune cells, thereby actively contributing to the development of innate and adaptive immunity. This is what, this is the macrophage. If this macrophage activated by our peptide, it could lead to all this production of interleukin 6. I did interleukin 6, TNF alpha, and IL-12 and IL-10. But this, like IL-12, can activate natural killer cells and also induce differentiation of CD4 cells. This is the most important for us here for chlamydia, because if the peptide can induce macrophage to produce IL-12, interleukin 12 is very important for activating the adaptive immunity, especially Th1. And Th1 is very important for chlamydia immune system. Okay, this is the method we use. We use the RMOMP. Then we have our macrophage cell, we call it the cell line J774. We use LPS as a control, and we have RMOMP. Then we stimulate the cell, 1 million cells for 24 hours. Then we collect supernatant, then we run ELISA. Okay, this study were conducted in mouse J774 macrophages. We did biochemical characterization. We did concentration kinetics or dose response type of study, we did time kinetics. We're just trying to see at what time any cytokines will be produced. Then the last part, we did the mechanism. Okay, the result. Here, this is the result of the first type of biochemical study. We call it effect of polymyxin. This, a lot of researchers, they do this because polymyxin B is antibiotics. Antibiotics, here, what we are trying to, from this study, what our goal from this? Our goal from this study, a lot of people, if they ask you, okay, you clone this and you did all this in bacteria, okay, maybe your peptide is contaminated with LPS from bacteria. And LPS, or lipopolysaccharide, is well known, a potent activator of our immune system. If your peptide is not pure, because we have done all this work in bacteria, we cannot say that what we have been seeing is due to our peptide. It could be due to LPS. That's why we have to prove that this is not due to LPS. We did this polymyxin B. Polymyxin B is an antibiotics well known used for a gram-negative type of bacteria by the mechanism of polymyxin by taking LPS out, bind to LPS and get LPS out of any type of system. When we use here, if you see, when we add our LPS to the culture and here, this is our peptide. We call it RMOMP 187, and this RMOMP plus polymyxin B. Here is showing there is no significant difference between these two. What that tell us? This is tell us that our peptide doesn't have any polymyxin. But what we did, we even after we clone and we get the peptide, we run it through a system, agarose system, that cleaning LPS. We did that because if the peptide, we take it surely, if we take it directly without cleaning it, and we did that. We did that and we saw very high type of response. Then we said that could likely due to LPS. Then we have to go through cleaning the peptide by using a saccharose type of column, type of containing this also polymyxin B, that cleaning LPS out of the peptide or the protein. We did that. We clean it, then we run this study. This is comparison because LPS here like a control for us. When we use polymyxin B with LPS, you see, LPS, reducing the production of this cytokines, interleukin 6. This is just showing us, telling us this what in the literature, and we also have seen that TNF alpha produce very high at 8 hours, after 8 hours of time, and also interleukin 10. While interleukin 6, it has this type of sigmoidal type of pattern. 24, again, then go to 72. While interleukin 12, we get it high at 24 hours. Okay, here, we try this in J774 cell line is a mouse cell line. Okay, we ask a question. If this peptide inducing mouse cells, is it going to do the same in human cells? If in future we try, to go into human type of study. We try this into human cell lines. We call it THP1. And also, this peptide inducing also human cell lines to produce very high of these important cytokines. And this time kinetics also telling us the same thing, is that sigmoidal type, of pattern after 24, then it will come to 48, then, while the cell by itself doesn't do anything. This for interleukin 6, also doing the same thing with human cells, is inducing, very strong immune response in human cells. Okay, the second part here, okay, some reviewers, they will ask you, you depend on all your work on cytokines. We want you to prove for us using a different techniques. Not just this, you will get this a lot if you go to your research or, It's not good to depend on one technique to prove something. Always try to get another type of technique to support your data. Here we did immunofluorescence to get support this data. Whether through immunofluorescence we can get also expression of this, protein or not. Here we use immunofluorescence. All of you know what immunofluorescence. Immunofluorescence is microscopic techniques depend on antibody that has a fluorescence dye. If that antibody bind to that antigen is going to, make a fluoresce, then you can easily visualize it under the microscope. Here, this interleukin 6, we use what dye called PE. PE is a red dye, and conjugate it with antibody to interleukin 6. And we use FITC dye that conjugated to interleukin 12 antibody. And we're able to see this clearly. This is a support that our, cell, this is the unstimulated here. This the RMOMP is stimulated. Unstimulated didn't show any type, of this interleukin. We did another technique to support this. We did flow cytometer or flow cytometry study. Flow cytometry study is a type of technique. Again, it depends on antibody-antigen interaction, but also has that antibody has to have some fluorescence. But here, flow cytometer is more reliable than even because here it will count for you exactly the number of the cells that showing this antigen-antibody interaction. Here again, we use 5 microgram of our RMOMP. This is the three different color. The red one is isotype. Isotype is antibody but cannot bind to the antigen because some of their epitopes is just modified. They cannot. While the green is unstimulated, just the cells, we didn't put any peptide there. While the third one, the blue, is the one that we put our peptide. This shifting, it will show that there is a stimulation. If this, you have to put this in a software, then the software will help you to analyze this data. Here is very clearly through another technique that our peptide is inducing expression of these cytokines. This for interleukin 6, and this is the same study for interleukin 12. These three different studies, they prove for us that now nobody can challenge us, telling us that no, this peptide is not doing anything. We know that the peptide can induce in vitro type of through different techniques. Now the other question again came because, as I told you, we look at the literature. We didn't see a lot of mechanism study. Okay, we said, okay, let's go ahead and do the mechanism of this peptide. Although it's part of MOMP, but still it will give us hints about how whole MOMP is working. What we did here, we are trying to know how this RMOMP inducing this cytokine secretion or production. If you know the story, this is our model. The whole model for the mechanism study. We think that this peptide, we call it RMOMP 187, is going to bind, as soon as get to our immune system, is going to bind to the receptor on the macrophage cells. We call them toll-like receptors. As soon as bind to toll-like receptors, is going to initiate a type of cascade of signaling pathway. This TLR is going to, we have done this in immunology, people, they know this. TLR is going to, immediately, if it binds to our peptide, TLR has, a type of domains. One is extracellular outside, the other one is inside our cell. TLR is going to change shape and conformation, especially here, intracellular. When change shape, as soon as being activated, now there is a molecule here called adapter molecule. We call it MyD88. MyD88 mostly is involved here as adapter molecule. It will come and bind here. And then, as soon as MyD88 have been activated, MyD88 is going to initiate a cascade of reaction. This is our model. We think that the MOMP is going to activate a TLR2 or 4. Because TLR2 or 4, according to chlamydia work, as chlamydia as bacteria, some research is saying that chlamydia goes through TLR2 and 4. Another one, he said TLR6 also is involved. Now we have a total of 13 TLR so far discovered. In human, about 9 of them, when we know their, how they work. Okay, this is our model. This, if we activate this TLR, is going to activate a cascade of reaction. We call it MAP kinase pathway. Then MAP kinase is going to activate NF-kappa B. NF-kappa B is a transcription factor. Has to move from the cytoplasm into the nucleus to bind to our gene here, and then start transcribing that gene to produce for us these cytokines we have just talked about. This is our model for the mechanism. Okay, what we did, toll-like receptors is a general information. Toll-like receptors are family of, a type of receptor found in some of our innate cells, like macrophage, dendritic cell, neutrophils. They have this receptor, they use it for recognizing foreign antigens to differentiate our own type of, cell from non-self, found in a lot of, especially dendritic cell, macrophage, and neutrophils. This group of toll-like receptors, they are responsible for detection this pathogen-associated pattern, we call it PAMPs. To date, we said over 13 TLR have been reported in human and mouse. But in human, we know 9. We know how they work. Of the 13 currently described, TLR 2 and 4 have been studied, especially in chlamydia. 2 and 4. 6, they said, is involved. That's why I focus in 2 and 4. TLRs, what they do, they activate NF-kappa B pathway. NF-kappa B has be translocated from the cytoplasm because it's transcription factor. And it has to go and bind to the promoter and start initiating gene expression there to produce the protein. And it goes to regulate cytokine expression through these adapters. The most important adapter and well-known is MyD88. And then TERA, MAL, and TRIF, these are other also type of adapters. Activation of NF-kappa B pathway is going to link for us innate and adaptive immunity. And this is very important for intracellular vaccine. You have to get a way of activating the adaptive immunity. Through this, if we're able to activate NF-kappa B, we'll be able to produce this cytokine, then link, and we can activate the adaptive immunity. I did all this with CD8 and CD86. The mechanism we are trying is just part of it, is not all this. Here, we think that our peptide is going to go here to TLR, then activate TLR, then go and activate MyD88. MyD88 is going to also activate this pathway of MAP kinase pathway. Then MAP kinase pathway is going to end here in NF-kappa B. Then NF-kappa B is going to translocate and start expressing these genes to produce these cytokines for us. Then these cytokines, they can do all this. They can, if the cell is intracellular and is infected, then apoptosis can take place, can kill, the cells. Or a direct antimicrobial response also. Some of these cytokines, they can kill the bacteria or any by themselves without any need. Okay, the important, they influence the adaptive immunity. This is very important. Okay, what method we use? For expression of TLR and CD, we work on CD80, CD86, CD40. We use here the flow cytometer. Again, we activate and unactivate. You have control, you have your peptide. Then after you add all this with antibodies, they have some fluorescence dye on it. Then, okay. We have only 3 minutes, but we didn't finish the second part of the story, the nanoparticle. But here, what we are able to do, we're able to recognize, and we have now the whole story of the peptide. We know that the peptide now, it goes through toll-like receptor 2. It's going to activate 3 of MAP kinases, that P38 and GNK. And we know that also goes through NF-kappa B. Because when we inhibit NF-kappa B, we have not seen any cytokines production. In these 2 minutes, I want just to go to the second part of this very quick here. Please, I want to show you just something about the nanoparticle, because this is very important part of this project. Nanoparticle, as most of you know, why we use nanoparticle, and why people now, every scientist is, going to, include nanoparticle in series. The nanoparticle is a new type of nanomedicine, is new type of research. Here we are using it for delivery system. Because chlamydia, one of the major problems of chlamydia is to target, to deliver. If you get the antigen, very strong antigen, can induce immune response. If you're able to target the tissue where that chlamydia mostly, being localized, you will be able to solve most of the problem. That's why we select nano. We are we don't want to see we are, but we are the first to do this and to get a paper out of this work on chlamydia using nanoparticle as delivery system. But this is just telling you a comparison what nano means. Nano means a small particle that we cannot visualize even using a microscope. We have to go to a type of atomic or electronic type of microscope before we see. It's when you compare it to all this, it's somewhere here, 10 to power -9 of the meter if you want to go. I use this nanoparticle called PLGA, polyelectric glycolic acid. I use it because of 4 or 5. First of all, is FDA approved for even human use. Secondly, nobody used it before for, to develop vaccine for MOMP. Thirdly, it's biodegradable. As soon as I take it, it will be degraded into lactic acid and glycolic acid. And these two components are part of our metabolic pathway. They can easily go into glycolysis and other part of pathway. That's why I use this and have very minimal type of toxicity. This is the structure, and this the nanoparticle. This is the method we use to encapsulate and get the nanoparticle. First of all, you have to we use this organic solvent called dichloromethane as our solvent to dissolve the polylactic glycolic acid. We use 5%. We encapsulate the peptide. Then we normally sonicate for 2 minutes. Then we get the ferrous emulsion, or we call it the primary emulsion. The second part, we add 1% PVA, polyvinyl alcohol. You know that this water and oil, dichloromethane is an solvent, organic solvent, and our peptide in liquid, they cannot mix together. We have to mix them through emulsifier. We use here polyvinyl alcohol, 1%, to allow our peptide to go inside the PLGA, the nanoparticle. Then at the end, we get this nanoparticle, and we achieve very high encapsulation efficiency. We get above 90% of our peptide able to be encapsulated inside. We have some techniques. It will tell you that what your encapsulation efficiency. Okay, here, this is how we do the encapsulation, and we get above 90. This is the characterization of the nanoparticle we did. You have to do this, to prove that, first of all, you have the nanoparticle, and also to give you an idea about the morphology and the structure of the nanoparticle. We did atomic force microscopy, scanning electron microscopy. We did FTIR, DSC, in vitro release, cytotoxicity, zeta sizer, zeta potential, and immunofluorescence microscopy. We characterize the peptide using all this technique, and we have our result here. This is the result of the AFM, or atomic force microscopy. It's just telling you how this structure. This is the peptide, our peptide, with before encapsulating it. It has a range of 20 nanometer. That's what we have got. Then we compare the encapsulated and uncapsulated. This, the PLGA by itself, give us this structure morphology and this size approximately here. If we get to compare the color here of this nanoparticle with this scale. And this, the nanoparticle after encapsulation. You see here, the size is increasing. After we put the peptide, the size. This is an indication that we encapsulate also. Another structure, we did also SEM in Auburn, because this the reviewer, one of the reviewer, he said, okay, this AFM, you get to provide us another type of techniques. Then luckily, we have this. We did it, but we didn't, give it to them at the beginning. They said, okay. When we sent, they said it's okay. But these are two different type of scale. This are very, high scale than this. Okay, this, the FTIR. FTIR study, it will just simply prove for you whether you encapsulate or not, because you are going to compare functional groups. Here, if you see here, we have three type of color. The red one is our PLGA by itself. The pink one is the PLGA with our peptide together, while the blue one is our protein. Any shift in these peaks is telling you that there is some interaction taking place, between. Because if you see here, the PLGA by itself is the red one. If we don't have any type of encapsulation, at least after we encapsulate, we're supposed to see all these peaks on the same, place with the PLGA alone. But if you see, you will see a lot of shift here. For example, this carbonyl functional group. Carbonyl gives us always this peak of 700. If you see there is a shift here when we encapsulate to 700, this shift is an indication that there is some interaction taking place between our peptide and the nanoparticle. And another also shift is taking place here. This, I think this is CC for chemistry people of a group, also showing some shifting here. And the most important functional group, if you see here PLGA doesn't have this group. Doesn't have that more of it. This, an OH group, mostly you find it in, structures that have a lot of OH hydroxyl group. And here, our peptide, our protein has a lot of is a peptide, has a lot of amino acids. That means a lot of OH group. And also after we encapsulate, we see this. That means this telling us, Okay, there is OH group, that means there is protein here. There is carbonyl group here, that means there is PLGA here. This spectroscopic or FTIR is just telling us that we are having the peptide, within or, in the nanoparticle. Okay, this release is very important. This is one of the objectives of using nanoparticle. Because for vaccine, why we use nanoparticle? We use nanoparticle for two things, for general vaccine, development. The first thing, for using nanoparticle is small in size. It can easily target any tissue and initiate a type of immune response than using, especially if you are working with peptide. Secondly, the nanoparticle is going to protect the peptide because, our system has a lot of proteases. Proteases are enzymes found everywhere in our cell that can degrade any type of, vaccine, especially peptide or subunit vaccine. That means another advantages for nanoparticle is protecting our peptide. But another most important advantage is the release. The nanoparticle is going to release this peptide in a very long time, That means that peptide is going to be seen by our immune system for a long time. And that helps our immune system, to be activated for a long time. And this has a very good advantage. Maybe in future, if you develop vaccine based on nanoparticle, we could alleviate that, the multiple shots for kids come after 6 months, after 2 months, after, it could also help on that. This all is advantages of using nanoparticle. This is here toxicity, telling us that our nanoparticle is not toxic. At highest concentration we use is still more than 80% of the cells are viable. This is the characterization I just told you about the size and zeta potential. Size is telling us our size is around 100, very good. And zeta potential is telling you the stability of your nanoparticle. If your zeta potential is high, that means your nanoparticle cannot clump, because zeta potential is just measuring the charge on surface. It will tell you how much charge you have. If you have too much charge, whether positive or negative doesn't matter. Just you need a high. The highest thing of zeta potential, they said around 27, 28. Our own give us 24. 24, that means we expect this nanoparticle is not going to clump. It's going to be dispersed and easily can, release that peptide and initiate type of response. This is just in a different color if that is not clear, the same type of study. We did also DSC. DSC, it will give you information about your, this nanoparticle. It's going to be stable at room temperature. At what temperature? If I want to transport this vaccine to Africa or Asia, I know that it will take some time, and the heat and the temperature is high. Is it going to be stable at that? Still, this giving us. In conclusion, we able to, to produce a peptide, and we able that peptide to show very good type of immune response. We able also to encapsulate that and test this encapsulated peptide on macrophages, and we able to publish that part. The later part, we publish it, but the beginning part, we couldn't publish it, but we finished the whole story, and we are ready to send this for publication. Now, our future to test the peptide and the nanoparticle on mice. Thank you so much for your time, and sorry for prolonging the talk. And if you have any question, I will be able to